Dear students, today we are going to learn organs of immune system. The organs and the associated tissues of immune system are categorized into three types: the primary lymphoid organs, the secondary lymphoid organs, and the tertiary lymphoid tissues. The primary lymphoid organs are also known as central lymphoid organs. Examples are thymus and bone marrow. The maturation of lymphocytes takes place at the primary lymphoid organs. The secondary lymphoid organs, they are also known as the peripheral lymphoid organs. Examples for secondary lymphoid organs are lymph nodes, spleen, and MALT. MALT is mucosal associated lymphoid tissue. The secondary lymphoid organs trap antigen and provide sites for the maturation of lymphocytes by providing the site for the interaction of these lymphocytes with the trapped antigens. The third type is the tertiary lymphoid tissue. Examples are CALT or cutaneous associated lymphoid tissue. The tertiary lymphoid tissues contain very fewer lymphoid cells and they are generally involved in inflammatory responses. This image shows the location of various organs and tissues of immune system. Coming to primary lymphoid organs, we know there are two primary lymphoid organs, thymus and bone marrow. The immature lymphocytes, either the T lymphocyte or the B lymphocyte, will become mature at the primary lymphoid organ. The T lymphocyte at the thymus and the B lymphocyte at the bone marrow. So, thymus. Thymus is a site of T cell development and maturation. Its maximum size will be reached during puberty. Its size is 70 gram in infants and 3 gram in adults. Thymus is a site of T cell maturation. So, during this process, 95 to 99% of T cells die in thymus due to either their self-reactivity or no reactivity to antigens. So only the T lymphocytes which are not self-reactive and which possess reactivity to foreign antigens will be selected and matured in the thymus. Structurally, thymus is a bilobed organ and it is located above the heart. Thymus consists of cortex and medulla. This bilobed organ is having a capsule and each lobe is divided into lobules. The lobules are separated from each other by strands of connective tissue known as trabecula. And each lobule is organized into different components. The outer compartment of the lobule is known as cortex and the inner compartment of the lobule is known as medulla. The cortex is densely packed with thymocytes or immature T cells, while the inner compartment or the medulla is having very low number of thymocytes. Thymocytes are immature T lymphocytes. So, thymus have the interior medulla and the exterior cortex, and there are several thymocytes or immature T lymphocytes. The thymus contains certain types of cells known as nurse cells. Nurse cells are thymic epithelial cells present in the outer cortex area. These nurse cells have long membranous extensions so that each nurse cell can be in contact with several thymocytes. The nerve cells can be in contact with up to 50 thymocytes. There are other cortical epithelial cells also, and they can also interact with numerous thymocytes at the same time. What are the functions of thymus? The main function of the thymus is to allow the maturation of T lymphocytes so that they can interact with foreign antigens and protect our body from infections. During the maturation of thymocytes or the immature T lymphocytes, they develop T cell receptors. Only those T lymphocytes which are capable of recognizing foreign antigens in association with MHC or major histocompatibility complex will be allowed to mature in the thymus. All those thymocytes which are not capable of recognizing the foreign antigens will be induced to death. Also, the T lymphocytes which are interacting with the self antigens will also be induced to death in the thymus. So, more than 95% of all thymocytes die in the thymus. Only 5% of the thymocytes will get mature to become the final T lymphocytes. So, thymus acts as a site for T cell selection. It is a site of maturation for T lymphocytes. Second primary lymphoid organ is bone marrow. Bone marrow is the site of B cell origin and development in human beings and mice. In birds, the B cell origin and development occur in a particular organ known as the bursa of Fabricius. While in cattle and sheep, the B cell origin and development occur in Peyer's patch. It is present in the wall of the intestine. Bone marrow is less defined and structured than thymus. The functions of bone marrow include hematopoiesis or the formation of blood cells. Bone marrow is the site of B cell maturation and also the B cell selection. Bone marrow contains several stromal cells which secrete various cytokines which helps in the maturation of B lymphocytes. During B cell selection, the B cells which are having a self-reactivity will be eliminated or will be induced to undergo death. Before going to the secondary lymphoid organs, let us discuss the lymphatic system. What is the lymphatic system? It is a system of vascular network of tubules and ducts that collect, filter and return lymph to the blood circulation. Lymph is a clear fluid that comes from blood plasma. It contains water, proteins, salts, lipids, white blood cells, etc. So how lymph is formed? It is formed from blood. As blood circulates under pressure, its fluid component or plasma seeps through the thin wall of the capillaries into the surrounding tissue. This fluid is known as the interstitial fluid, and a major portion of this fluid will return back to the blood through the capillary membranes, and the remainder portion is known as the lymph. It will flow into a network of tiny lymphatic capillaries. From these tiny lymphatic capillaries, the lymph will flow through a progressively larger collecting vessels known as the lymphatic vessels, and ultimately it will reach the lymph node. The flow of lymph through the lymphatic vessels are due to the muscle contractions, the movements of the body's muscles. Also, there are a series of one-way valves along the lymphatic vessels which ensures that the lymph flows only in one direction. Lymph flows through the tiny lymphatic capillaries, and then they flow through a series of larger collecting vessels known as the lymphatic vessels. The afferent lymphatic vessels reach the lymph into the lymph node. Lymph node is an organized lymphoid tissue, and the lymph leave the lymph nodes through efferent lymphatic vessels, which drain the lymph into the circulatory system, to the blood system, through the thoracic duct or the right lymphatic duct. The largest lymphatic vessel is the thoracic duct, and this thoracic duct empties the lymph into the left subclavian vein near heart. So, what are the functions of the lymphoid system or the lymphatic system? It drains and returns the interstitial fluid back to blood. It absorbs and returns lipids from the digestive system to the blood. It also filters the fluid of pathogens, damaged cells, cellular debris, cancerous cells, etc. Now we are going to discuss the secondary lymphoid organs. The secondary lymphoid organs are lymph node, spleen, and mucosal associated lymphoid tissue or MALT. These secondary lymphoid organs contain lymphoid follicles, primary follicles and secondary follicles. What is a primary lymphoid follicle? It is a network of follicular dendritic cells and small B lymphocytes. When a primary lymphoid follicle encounters an antigen, it will become a secondary follicle. A secondary follicle contains more number of lymphoid cells. There will be larger numbers of proliferating B lymphocytes, non-dividing B lymphocytes, helper T cells, macrophages, and follicular dendritic cells. So, lymph node, spleen, and MALT, they all contain primary lymphoid follicles and secondary lymphoid follicles, generally surrounded by a fibrous capsule. Coming to the lymph nodes are encapsulated bean-shaped structures containing lymphocytes, macrophages, and dendritic cells in a reticular network. The lymph nodes are present at junctions of the lymphatic vessels. They act as filters for antigens. They are the first organized lymphoid structure which will encounter antigens. The lymph enters into the lymph node through several afferent lymphatics, and the lymph will leave the lymph node through one efferent lymphatic. The antigens which are coming through the lymph will get trapped in the lymph node through the cellular network of phagocytic cells and dendritic cells. Morphologically, a lymph node have three distinct parts: the cortex, the paracortex, and the medulla. In a lymph node, the cortex is the outermost layer and the innermost layer is medulla, and the layer paracortex lies between medulla and cortex. The outermost layer or cortex contains lymphocytes, mainly B lymphocytes, macrophages, and follicular dendritic cells, which are arranged in a primary follicle. When an antigen enters the lymph node, the primary follicles in the cortex becomes a secondary follicle. Under the cortex is the paracortex. Paracortex mainly contains T lymphocytes and interdigitating dendritic cells. These interdigitating dendritic cells are highly capable of antigen presentation to activate the T helper cells. The innermost layer of a lymph node is the medulla. Medulla contains very less number of lymphoid cells and it contains mainly plasma cells. Plasma cells are a type of B lymphocytes which are capable of secreting antibody molecules. So in a lymph node, there are several afferent lymphatic vessels which empty or which carry lymph into the lymphatic node. Lymph coming from the tissues through the afferent lymphatic vessels will flow through the cortex, paracortex and medulla, and it will finally reach the efferent lymphatic vessel. During this travel, any antigen or bacteria or microorganisms which are carried through the lymph will get trapped within the lymph node by the action of phagocytic cells and dendritic cells. These cells will get activated, they will multiply and elicit an immune reaction so that the lymph leaving the node will be having higher concentrations of antibodies and lymphocytes. The antigenic stimulation in a lymph node will increase the concentration of lymphocytes inside the lymph node. This will result in the size increase of the lymph node. So the lymph node will get swelled, resulting in a visible increase in its size. So the lymph nodes are specialized for trapping antigens or microorganisms that get entered into a body through local sites, through skin or through mucous membrane, etc. The next secondary lymphoid organ is spleen. It is a large ovoid organ situated in the left abdominal cavity. Spleen functions in filtering blood-borne pathogens and antigens. Structure-wise, spleen is surrounded by a capsule that extends a number of projections into the interior of the spleen. The projections are known as trabecula, and the trabecula gives spleen a compartmentalized structure. There are two types of compartments, the red pulp and the white pulp. A diffuse marginal zone is there between the red pulp and the white pulp. Spleen contains several different types of immune cells such as lymphocytes, macrophages, dendritic cells, and red blood cells or RBCs. The main functions of spleen is to filter out older RBCs, to trap and respond to antigens in the blood circulatory system, and spleen is a site where activated B cells against the antigens will be produced. So, the spleen is surrounded by a capsule. The capsule extends several projections known as trabeculae into the interior, and this trabeculae gives spleen a compartmentalized structure. The spleen have two compartments, the red pulp and the white pulp. There is a diffuse marginal zone between the red pulp and the white pulp. The red pulp of the spleen contains a network of sinusoids. This area contains macrophages, red blood cells, and few lymphocytes. This is the area where old and defective RBCs are destroyed. The white pulp area of the spleen, it surrounds the splenic artery and it forms pals or periarteriolar lymphoid sheath. Pals is mainly having a larger population of T lymphocytes. There are several primary lymphoid follicles in the periarteriolar lymphoid sheath or pals. These primary lymphoid follicles contain larger numbers of B lymphocytes. There are also lymphocytes and macrophages in the periarteriolar lymphoid sheath or pals. The blood-borne pathogens or antigens and the lymphocytes enter the spleen through the splenic artery and will reach the marginal zone, the area between the red pulp and the white pulp. In the marginal zone, the antigens will be trapped by dendritic cells known as interdigitating dendritic cells, which will carry the antigens to the pals region, the periarteriolar lymphoid sheath. Here, the dendritic cells will present the antigens to T helper cells, which will further activate B lymphocytes so that an immune reaction against the foreign antigen will be initiated. The next type of secondary lymphoid organ is the mucosal associated lymphoid tissue or MALT. These are present along the mucosal membranes of the digestive tract, respiratory tract and urogenital system. Since these mucosal membranes are the major sites of entry for most pathogens, these mucosal membranes have to be protected, and this immunological defense is mediated by a group of organized lymphoid tissues, and these lymphoid tissues are known as the MALT. MALT contains large numbers of T lymphocytes, B lymphocytes and macrophages, and they encounter antigens passing through the mucosal epithelium. There are a special type of cells known as M cells or microfold cells in the case of intestinal MALT. Microfold cells or M cells are present in the gut-associated lymphoid tissue of the Peyer's patch and in other parts of the gastrointestinal tract. These are a type of cells which have the ability to take up antigens from the small intestine via different mechanisms such as endocytosis, phagocytosis or transcytosis. Then the antigens will be given to antigen presenting cells so that an immune reaction will be elicited. These mucosal associated lymphoid tissues or MALT are subcategorized into several different types based on their location. They are GALT or gut associated lymphoid tissue, BALT or bronchus associated lymphoid tissue, NALT or nasal associated, LALT or larynx associated, SALT or skin associated, CALT or conjunctiva associated, or LDALT, which is lacrimal duct associated lymphoid tissue. So these tissues may vary in their organization. They may be either barely or loosely organized clusters, or there may be well organized structures. Examples for loose or barely organized clusters are the lymphoid cells in the lamina propria of intestinal villi, which is an example for GALT. Examples for well organized structures are tonsils, appendix, Peyer's patch, etc. Peyer's patch also comes under GALT or gut associated lymphoid tissue. This diagram shows the anatomical locations of mucosal associated lymphoid tissue or MALT. The tonsils is an example of MALT. The tonsils are found in three locations: lingual at the base of the tongue, palatine at the sides of the back of the mouth, and pharyngeal in the roof of the nasopharynx. Tonsils contain lymphocytes, macrophages, granulocytes, and mast cells. The cells in the tonsils trap and act against antigens entering through the nasal and oral epithelial roots. The tertiary lymphoid tissue is a cutaneous associated lymphoid tissue. It is present in the epidermal layer or outer layer of the skin. The epidermal layer of the skin contains keratinocytes, Langerhans cells, and intraepidermal lymphocytes. Keratinocytes are specialized epithelial cells which are capable of secreting a number of cytokines. These cells express class II MHC molecules, thereby functioning as antigen-presenting cells. So the keratinocytes can elicit a local inflammatory reaction upon antigen entry. The Langerhans cells are also antigen-presenting cells. They can engulf or internalize antigen by phagocytosis or endocytosis and can present the antigen to T helper cells, thereby activating them. The epidermis also contains intraepidermal lymphocytes. So, we discussed about the organs involved in the immune system. The primary lymphoid organs, thymus and bone marrow. The secondary lymphoid organs, lymph node, spleen, and mucosal associated lymphoid tissue. And the tertiary lymphoid tissue, that is a cutaneous associated lymphoid tissue. Hope the class was clear to you all. Thank you so much for listening.